Okay, it just hit about 1:33 on my watch, UK time. Good afternoon, everyone from everywhere in the world. I can see different people from different countries that I recognize. So thank you so much for joining us. And welcome to the fourth edition of the Genomic Surveillance Unit GSLU Malaria Journal Club in the monthly series. Today's webinar is the first vector-focused one. The other three, which are on YouTube, were focused on the parasites field. My name is Babalo Chauke, and just to give you a few ground rules, the Journal Club is being recorded and will be available on our YouTube channel, the Malaria Gen YouTube channel, either today or tomorrow. So you can rewatch today's presentation and share with your colleagues as well. Please stay on mute. We will have a Q&A session at the end where you can raise your hand or type your question or comments in the chat or Q&A box. Without further ado, I would like to introduce the speaker for today, who is Dr. Eric Lucas. The title of the talk, as you can see on the slides, is the title of the newly published paper, and it's titled Genome-Wide Association Studies Reveal Novel Loci Associated with Pyrethroid and Organophosphate Resistance in Anopheles gambi, as well as Anopheles coluti. Just a quick bio about Dr. Eric Lucas. Dr. Eric Lucas is a senior research associate from the Liverpool School of Tropical Medicine, and he's working on the genomics of insecticide resistance in malaria mosquitoes. Dr. Lucas uses the whole genome sequencing data to understand the genetics of resistance, its evolution, and how best to design genetic screening methods for resistance diagnostics. Dr. Lucas, thank you so much for joining us. You may begin your talk.
Thank you so much, Babalo. Thank you very much for inviting me. It's a pleasure to be here. Yes, so as Babalo said, I'm going to give you an overview of the paper we published a few months ago on genome-wide association studies looking to understand the genetic associations with insecticide resistance in malaria mosquitoes. So apologies for the bare nature of the first couple of slides. I realized at the last minute that probably not everyone in the audience was familiar with malaria and vectors, but so malaria kills around 600,000 people every year across the world, but primarily in Africa. And the primary means of malaria control is still based on insecticides. So although there's a bunch of other methods being developed that are very promising, we're still very reliant on insecticides to kill the mosquitoes that transmit malaria as the primary mode of fighting malaria. And this has been very successful, but this success is threatened by the evolution of insecticide resistance in mosquitoes. So in the same way as we're seeing evolution of resistance to anti-malarial drugs in the parasites, we're also seeing evolution of resistance to insecticides in the mosquitoes that carry malaria. And so good malaria control, a good malaria control program would involve choosing the best insecticide to use in a given area, given the compounds that the local mosquitoes are resistant to. But how do you do this? How do you determine what the local mosquitoes are likely to be susceptible or resistant to? And it's beyond the scope of this to go into all of the different methods that you could use and their pros and their cons. But we believe that a very promising way of doing this is genetic screening. So if we can find out what the genetics, what the mutations are that are associated with insecticide resistance, then we can very quickly and reasonably cheaply screen a population of mosquitoes and say something about what they are likely to be resistant to and what interventions are likely to be successful on that population. And all of this starts with identifying those mutations that are associated with resistance. And that's what this paper set out to do. So we were trying to investigate the genomic basis of insecticide resistance in mosquitoes. So we did this within something called the GUARD project, which is a collaboration between LSTM, Sanger, the University of Oxford, and partner institutions in both West and East Africa. And essentially, in all of these populations of mosquitoes, what we did was collected larvae from the field, raised the offspring in the lab, and performed bioassays in order to obtain distinct resistant and susceptible phenotypes. Now, traditionally, the way this is done is you take a load of mosquitoes, you expose them to a single dose of insecticides, You compare the genomics of the survivors and the ones that were killed by the insecticide. And you say that the survivors were resistant and the ones that were killed were susceptible, and you compare the genomics between the two. What we did in order, so the problem with this is that you can get quite a strong overlap. There's a lot of sort of randomness involved in whether a mosquito will be killed, and so you can end up with actually your so-called resistant pool is some resistant and a bit of everything else, and your susceptible pool is some susceptible and a bit of everything else. And so, in order to get stronger differentiation, to get increased power in our analyses, what we did was we exposed one group of mosquitoes to a high dose of insecticide that was previously determined to kill between sort of 80 to 90% of mosquitoes. And we exposed another group to a low dose of insecticide that we knew would kill about 10 to 20% of mosquitoes. And we took the survivors from the high dose and the dead from the low dose. And so this gives us a sort of the most resistant insecticides to compare to the least resistant, the most resistant mosquitoes, sorry, to compare to the least resistant mosquitoes, giving us a stronger contrast. And we collected all of these mosquitoes and submitted them to the Anopheles gambiae 1000 Genomes Project for sequencing, SNP calling, CNV calling, and haplotype phasing. And so we got back all of this genomic data, and the mission then was to try and find out what the genomic associations were with insecticide resistance. We did this with two insecticides, deltamethrin on the one hand, because it's a common constituent of bed nets, and primperos methyl, which has recently started to be used for indoor residual spraying as a non-pyrethroid, and for which we don't yet know all that much about the genetics of resistance. So as I said, we had populations from both East and Central Africa, but because of Nagoya difficulties, we got all of the data from West Africa well before we got the data from East Africa. And so this initial paper focused on the samples from West Africa because those are the ones that we had data for at the time. We're currently working on the data from Tanzania, and we hope to get to obtain the data from Kenya soon. And so these can sort of form additional data to complete the picture. So let's zoom in on that West African region where the samples for this paper came from. So we had samples from Côte d'Ivoire, Ghana, Togo, and Benin of both Anopheles gambiae SS and Anopheles coluzi. And these, after sequencing, after quality control, these are the numbers that we got back from the Anopheles gambiae 1000 Genomes Project. So these were slightly smaller than we had hoped because a lot of samples didn't pass sequencing QC. And you can already see that in one population in particular, the one in Aboisseau, which is the one from this area in southeastern Côte d'Ivoire, we had very, very few dead individuals, which means that we were able to say things about mutation frequencies and charts in the population, but this sample size was too small to do any association studies to ask what are the genomic associations of resistance. For the other samples, we initially had sample sizes ranging from the low 30s to the high 80s, but we had a few challenges. So obviously all mosquitoes were identified morphologically as females before we sent them for sequencing. Females are the sex that we're interested in because that's the sex that carries malaria. And so fundamentally, that's the sex that's being targeted in malaria control campaigns. A very small number turned out after sequencing, we could see from the genomic data that they were males. So just 4 mosquitoes turned out to be males. But we then calculated relatedness pairwise across all of our samples using the genomic data. And we found that a lot of the samples were actually siblings to each other. And if you're trying to do an association analysis, then the inclusion of siblings will confuse the results because essentially you have non-independence of data points. So 2 sibs might both be resistant and both share a given mutation, but those 2 things are not necessarily linked. They might just share that mutation because they share half of their genome. And then there we lost much larger numbers of samples. So we had to remove a lot of samples for being siblings, and the final sample size looked more like this. So we're going from sort of the mid 20s to the mid 80s here. So the next issue we had was that we found out that we had problems with bacterial contamination in our samples. So this started off. So the first thing we did with our data was do what's called a genome-wide association study, and essentially we calculated for every single mutation in the genome, we calculated the association between that SNP's genotype and resistance. So whether the mosquitoes that carried them were dead and alive. And so for every single SNP in the genome, we get a p-value, and you can plot those p-values across the genome, and uh, and then on the y-axis you can put minus log of the p-value, and this is called a Manhattan plot. And basically, highly significant SNPs will stand out as being sort of very high on this plot. And so we got very excited initially, that this is a Manhattan plot from one of our samples, and we see this huge sort of column of highly significant p-values in this region. We started digging into it. We started asking what it was. But one of the things we found when we dug into it is we looked at the actual alignment data, which looks like this. So if you've not seen, this is an IGV plot. If you've not seen this before, I'll walk you through it. Essentially, this shows all of the reads, all of the sequencing reads in the data aligned to the reference genome, where each read sits along the genome. And what you should look at is this top bit of the plot here, which is the total coverage. So the number of reads that align at every position. And you expect this to be roughly uniform across the genome. You don't expect certain areas of the genome to have disproportionately more reads than others. And what you see is that you have the baseline over here, sort of even coverage, and then you get these regions, in this area where we had these lots and lots of significant p-values, you get these huge regions of massively increased coverage, and that's quite suspicious. That suggests that we have reads coming from elsewhere that aren't part of the mosquito genome. And this was already after alignment to Anopheles gambi. So you'd hope that a lot of non-gambi reads wouldn't align, but in this case they did. And if we took these sort of superfluous reads and we assembled them and we blasted that, the reads tended to hit a species of bacteria called a sire. The other suspicious thing was that all of these, every sample that had these mutations suggested in this region were heterozygote for those mutations, but the mutation was fairly frequent. But we never found homozygote mutants, and that's unusual. So if you find a mutation that's at reasonably high frequency in the population, you would expect to find at least some homozygotes for that mutation, and we weren't seeing any. And this is another suggestion of contamination, because it suggests that all samples that have the mutation, that mutation is just just there because of some extra reads that are coming from elsewhere. So you have the mutation coming from the extra reads, and you have the wild type allele coming from the actual reads, and so you consistently get heterozygotes. And so we used a metagenomics pipeline to just calculate what proportion of reads in the sample came from a sire. And if we compared the samples that were wild type at these loci to the ones that were heterozygote at these loci, the heterozygote, both amongst the dead and amongst the alive samples, the heterozygote samples had much, much higher levels of sire reads in them. So again, this was indicating that these mutations are the result of bacteria. So this still was potentially quite a promising story, because it could just be metagenomic resistance. So you could argue that perhaps the presence of these bacteria were providing resistance to the mosquitoes that carried them, and that's why we were seeing this association. But if we then mapped the amount of Asaya contamination to the wells that the samples came from, so this is just an example, one of our sample sites, we have one plate that had the dead individuals in them. We had one plate that had the alive individuals in them. The size of each circle and the darkness of the red corresponds to how much contamination from Asaya there was in each of the samples. And so you can basically see that all of the alive samples had a decent amount of contamination. The dead samples had a mixture, but it was very much dependent on where you were on the plate. So essentially the left-hand side tended to have very little contamination, and the right-hand side tended to have quite a lot. So the contamination is not, the bacterial reads are not present in the there's no reason to believe that the bacterial reads were present in the mosquito before they died, essentially. And so we had to control for this. Essentially, what we had to do was just remove all of the mutations that we found that were significantly correlated with the presence of a sire in the sample, which removed, of course, these beautiful peaks that we had got so hopeful about. So mostly including that as a note of caution, especially in genome-wide association studies, you really need to dig into your results. Don't take anything at face value. And if something is too good to be true, it very often is. And it's really worth sort of digging into your results and finding out really what's the cause of them. Okay, so we've now removed a lot of our samples that were siblings, removed a lot of the ones that were males. We've removed all of these SNPs that we believe to be associated with bacterial contamination. And then we repeated that process, essentially the Manhattan plot, performing a logistic regression of phenotype versus genotype at every single SNP in the genome. And we controlled for false discovery rate using the R package FDR tool. So what we found after this was that very few signals survive false discovery rate control. So in deltamethrin, we found no significant mutations in any of our populations. And for pyrimethos methyl, nearly everything is in ACE1, and ACE1 is a known resistance locus because ACE1 is the target site for PM. And it's known that there's a big gene duplication and associated SNP in ACE1 that is associated with insecticide resistance. And if we plot where are the significant SNPs, so this is essentially just a plot across the genome and in windows and the number of significant SNPs in each window, we basically see that nearly everything is in ACE1. And whilst there is a smattering elsewhere, this doesn't really mean much because essentially what false discovery rate control does is it says we accept a certain percentage of false positives in our data. So if we set the false discovery rate control to 1%, which is the case here, then we accept that 1% of our SNPs will be wrong. And so if you have thousands and thousands of SNPs coming from ACE1, then you end up with dozens and dozens of SNPs, 1% of those thousands that aren't actually truly associated with resistance. Okay, so that's if we just do a big agnostic sort of GWAS across the genome. We don't find much, but the false discovery rate control is absolutely brutal on this because there are millions and millions of SNPs. So what if we look at markers that a priori we expect to be important? So we just, instead of looking at millions and millions of SNPs, we just look at a few. So in deltamethrin, we know that KDR is the target site of that insecticide, so we expect to potentially find associations there. And we know from previous studies that copy number variants in CYP6AA1, so the number of copies of the gene of CYP6AA1 are both associated with resistance. We know this from previous studies in other populations. Now, the thing about KDR is that mutations in KDR are completely fixed in our data. So there are no wild type samples. There are no wild type alleles. However, what we do have is two different mutant alleles. So in Anopheles coluzi, there's the 402L plus 1527T haplotype that exists alongside the 995F haplotype. So these are two different resistant mutations. And what we found in our data when we just looked at this SNP in isolation, so no false discovery rate control, we found a marginally significant association. So we found that the 402L and 1527T haplotype provides higher resistance to deltamethrin than the 995F haplotype. In CYP6AA1, we looked at copy number again because we know from a population of Anopheles gambiae in Uganda that a CNV, so a copy number variant, a gene duplication in CYP6AA1 is associated with resistance there. We found elevated copy number of CYP6AA1 in Anopheles coluzi from this West African data. And in one of those locations, Korle-Bu, which is over here, we found that copy number of CYP6AA1 was again associated with resistance to deltamethrin with a P of 0.01. And there was actually a very similar trend in Abranku that wasn't quite significant. It was very close to significant, and it was in the same direction. And again, it was copy number of CYP6AA1 that was associated with resistance to deltamethrin. Now, in the Uganda data that I talked about before, there was one mutation, one CNV allele, we call them. So one duplication of CYP6AA1 that has spread across the country and that is bringing with it resistance to delta methrin. What we found in these West African data was there's not just one CNV. So this table is showing you for each of our populations all of the different CNV alleles that we know about from this study and others, and for each of those alleles, what the frequency was. And we can see, for example, in Anopheles coluti from Corlebu, which is where we found that significant association, there are 5 different CNV alleles that are mixing in the population. So it's not like in Uganda where we have one allele that's spreading. We have at least 5 different alleles here. And we found 3 of those 5 alleles, actually, and the 4th one in Abranku, and we found different CNV alleles in Anopheles gambiae, including, so this first column just shows whether there's any kind of CNV in case it's a new allele that we don't know about, and we found a reasonably high frequency allele in Medina, but we didn't find association with resistance. And actually this CNV covers other genes in that cluster, but it doesn't actually cover CYP6AA1. So association with resistance of copy number, but lots of different CNVs involved mixing in the same population. And then for PM, essentially what we found was that ACE1 was extremely significantly associated with resistance in 4 out of 5 of our data sets, and that didn't really come as a surprise. But the thing to note about these results and these p-values that we're getting is that apart from ACE1, where we're getting very small p-values, these other p-values that we found would not survive forced discovery rate control. So that's why these mutations are not coming out in our genome-wide association study, because a p-value of 0.01 is never going to survive the kind of forced discovery rate control that you have to apply to a collection of millions and millions of SNPs in order to make sure that you minimize your false positives. And so what this is telling us is that the kind of effect sizes that we see for resistance-associated mutation can't be detected with a GWAS of these sample sizes. And I'll get back to that later because we then did some calculations to work out, well, what sample size would we need? Given these effect sizes that we're seeing here, what sample size would we need in order to have detected these in a genome-wide association study? But in the meantime, let's continue exploring these data. So having looked at individual SNPs and having done this agnostic search across the genome for individual SNPs that are associated with resistance, we wanted to see if there was a more powerful method of looking for resistant SNPs or resistance regions in these data. And what we did was we again swept across the whole genome, but instead of doing every SNP independently, we group them into windows of SNPs. So we take the first 1,000 SNPs along the genome, then we take the next 1,000 SNPs, then we take the next 1,000 SNPs. And for every one of those windows, we calculate some statistic of interest. And the reason why this can be powerful in amplifying the signal is, first of all, you're reducing your sample size because you have 1,000. If you analyze things in groups of 1,000 SNPs, then you have 1,000 times fewer data points, and so your false discovery rate is less problematic. But also you're amplifying the signal because SNPs that are very close to each other in general are not independent. And so that window of 1,000 SNPs is probably telling you the same thing. Or all of those 1,000 SNPs are telling you roughly the same thing. And that will be particularly true for a window that has recently undergone positive selection, which is probably going to be the case of any window that's associated with insecticide resistance. So we're amplifying all of the signal, and we're particularly amplifying the signal in regions associated with resistance. And so hopefully that can make regions that that don't come out as significant when you do an individual SNP-wise analysis suddenly will stand out. So one example of a statistic that we can calculate is FST, okay? And that's a measure of population differentiation. So if you can calculate FST between one population and the other, and a high value of FST will tell you that those populations are very genetically different, and a value of FST close to 0 will tell you that those populations are not particularly different. And so we can compare our dead and our alive population using FST. And what we expect is that regions where there's no association with resistance will have an FST close to 0 because these are fundamentally actually all mosquitoes from the same population, and so they should be genetically identical. But regions where there is association with resistance, then through our bioassays, we've pulled out differences in genetics in those two groups, and they should stand out. So we can do that. We calculate FST. We plot it across the genome. This is in one population in particular. So Anopheles gambi from Begida exposed to deltamethrin. And we get values of FST plotted across the genome. And we want to identify peaks in this FST. There's no particularly hard and fast rule for how to do this. The way we did it was, so we used the fact, so negative FSTs should only be a result of noise. There's not really any biological reason why you should detect negative FST. And so we use the idea that the negative side of the distribution, if you start from the highest point of the distribution, the negative side should tell you something about what, so none of those should be biologically meaningful. They should just be due to noise. And so we use that to tell us something about how broad the distribution is for those regions that are just due to noise. So we take that distance, we take the same distance again from in the other direction, we multiply it by 3 in order to be conservative and to deal with the fact that the distribution of FST is not actually very symmetrical. And we use that as our detection limit. So we say any FST value greater than that, we call a peak. There are other methods that you can use to detect peaks to have pros and cons. This is the one that we used. So if we do that with the data I just showed you, we identify these 4 windows here as peaks in the data. So one suspicious thing here is that we're seeing significant hits in ACE1, and this is in the experiment exposing things to deltamethrin. And this is very surprising because we don't expect, ACE1 is the target site for organophosphates. We do not expect this association in pyrethroids. And this made us realize that there's another problem with using windowed statistics, FST or any other statistic. And it's one that's not really fully appreciated. I don't think I haven't really come across acknowledgement of this problem before. And essentially what it is that, as I said earlier, SNPs next to each other are telling you the same picture. And that means that, and this is particularly true in SNPs where there's been recent positive selection. So in a window of 1,000 SNPs, let's say that here we have, so this is, you're looking along the genome. This is a genomic window where you have 1,000 SNPs within them, and you calculate FST for that window. What that essentially means is calculate FST at every SNP and take the average. Now, these are relatively, I mean, they're still not completely independent because, as I said, they're close to each other, but they're relatively independent from each other because they're in a region that hasn't been under selection. There's been lots of time for recombination, lots of time for mixing. And so all of these SNPs are potentially telling you a slightly different story. And if there's random noise, they're all subjected to random noise in different directions and different amplitudes. And so even if your distribution, your null expectation of distribution of FST for a given SNP is quite wide, because of the law of averages, if you average these 1,000 SNPs, you get quite a narrow distribution of FST. So everything should be fairly close to 0. If you take a window that's under selection, where every SNP now has not really had time to undergo recombination, every SNP is much more likely to tell you exactly the same story. So if you take a SNP at random, then its FST will still be drawn from this rather broad distribution of FST. But the thing is that the next SNP along is probably going to, if this first SNP that you looked at happened to have by chance a high value of FST, the next SNP you look at probably will do as well, and the one after that, and the one after that. And so actually the law of averages then plays out a lot less over the 1,000 SNPs, and you end up with a distribution that's slightly narrower than the one for a single SNP, but not much, compared to the one that's much, much narrower in a region that's not under selection. And not only do you get this within a window, but you'll get the same thing with the next window along. So if this window happens to be high, then this window will probably be high as well. And so essentially what you get is that in regions where there's been very strong positive selection, you will get a much higher variance in FST. You could get very high FST just by chance, and they will stand out on a genome-wide plot next to all of those regions that are more like this, where the FST tends to be very close to 0. And so we could explore that. What we did was we randomized the phenotype labels for our FST calculation. So when we're calculating FST, what we do is we compare the dead individuals with the alive, and we say what's the genetic difference between them. And what we can do is we can randomize those groups. So we can just mix up those dead and alive labels. So we randomly allocate you. We're going to call you dead and you alive, even though you're not. and we recalculate FST between two random subsets of those samples. And if that high FST is due to true differentiation between dead and alive, then we should find that our observed FST is bigger than anything we see in our randomization. But if it's just wildly fluctuating, and you just very often get high FST by chance, then a good bunch of our simulations will end up having an FST that's just as high or even higher as the one that we actually observe. And so if we plot those randomizations, so this is now again across the genome, but instead of plotting one true value of FST, I'm plotting 1,000 randomized values of FST, 1,000 values of FST calculated after randomizing those phenotype labels. And you can see that in ACE1, there is a peak. So just in the randomized data, the values in ACE1 can take on higher values than the regions around it. And we can use this to apply a sort of statistical test to our peaks, because we can say, okay, we'll only accept a peak that is higher than 1% of our simulations. And if any peak that's lower than 1% of our simulations for that same window, we reject. And so if I overlay that plot of actual calculated FST from earlier, then suddenly our peaks in ACE1 become non-significant, because yes, they're high, but we get a lot of our simulations are at least as high. And so it's probably just due to chance, because there's a selective sweep here. Yes, there's a selective sweep here because there's been selection for resistance to PM. But know that sweep is not associated with resistance to delta methrin. Whereas these other peaks, which are a bit lower, we keep them because they're not as high as this one, but there's better evidence that they're actually higher than you'd expect on average for that genomic region. Okay, so one interesting finding that came out of this was that so this is now this FST plot in Anopheles gambiae from Obuasi exposed to delta methrin, but we see the same thing in Medina. So both of these populations show the same thing, which is a huge, very significant peak in FST around this CYP6AA1 region. So this was the region where in Anopheles coluzi, we found that copy number was associated with resistance. It's the same thing that's been found in Gambia and Uganda. But in Anopheles gambiae here, although there were CNVs, we did not find those CNVs to be associated with resistance. So copy number of that gene was not associated with resistance, but there is a strong signal that there is something going on in that region, some indication that there is association with resistance there. And what we can do is we can go in, we can find out what the haplotype under selection is in that region, and we can ask, is there an association between that haplotype and resistance? And the answer was yes, and with a p-value of 0.002. We can also ask whether that haplotype, so we found this in both Obuasi and Medina. We can ask whether the haplotype under selection is the same in Obuasi and Medina. So we did a haplotype clustering tree, and here all of the labels are colored so that alive is blue and orange is dead, but Medina have darker colors than Obuasi. And you have this big cluster under selection, and the samples here are shared. Okay, so we have samples from both Medina and Obuasi. So that main haplotype under selection is the same one in Medina and Obuasi. But it's not copy number variation, and we don't know what it is. But probably it's some other mutation that is increasing expression of CYP6AA1 or another gene in the cluster that is bringing about insecticide resistance. So we're consistently seeing the same gene, or the same gene cluster in CYP6AA1 being associated with resistance, but we're seeing multiple different mutations segregating in the same population. So lots of different CNVs, for example, in Corlebu. And we're seeing sometimes it's copy number variation and sometimes it's something else that's associated with resistance. So the summary so far is that resistance mutations don't have a strong enough effect to be detected with a GWAS, with the sample sizes we used here. And even at a locus where resistance is consistently found in different populations, we're seeing different mutations being important. So that was a sort of quick overview of how we did the genome-wide scans and a spotlight on one of the results that we found. Quick overview of sort of the rest of the results, if you like. We'd used a few different methods to do the genome-wide scans. So one was FST, and because FST isn't directional, you can have high FST, but you don't know which, you don't know whether the selection appears to have been, or you don't know which way the association goes. You don't know whether the selection seems to have, the selected haplotype is in the resistant or the dead or the susceptible samples. we essentially did what I just presented. So we calculated FST, and then whenever we found a significant peak in FST, we dug into it. We looked at the haplotype clusters within those windows, and we asked, are those haplotype clusters significantly associated with resistance, and if so, in which direction? So does having the haplotype cluster make you more resistant? And we identified those windows where having the cluster made you significantly more resistant. We also used a measure which we call delta H12. So H12 is a measure of selection because it's a measure of how common are individual separate haplotypes in the population. So if a population has a few very high frequency haplotypes, that suggests that those haplotypes have been selected. They're very beneficial to the mosquitoes. You get a high signal of H12. If you get a mixture of lots of different haplotypes in your population, then it suggests that no selection has happened, and you get a very low value of H12. So you can plot H12 across the genome, and what we did was we calculated H12 across the genome separately for dead individuals and for alive individuals, and we took the difference, with the idea being that in a region where there has been strong selection and where that selection is associated with resistance, then you will find that the alive individuals will be very dominated by that single haplotype, will have a high value of H12, whereas the dead individuals will have a mixture of other haplotypes, will have a very low signal of H12. And so you turn this signal of H12 into something that looks like this, that's symmetrical around 0, and we keep only positive peaks. So again, we do the simulations, as I said before, and we do the peak finding algorithm, but here we're keeping only the positive peaks, not the negative ones. We also use something called the population branch statistic, which is essentially a measure of how much evolution has happened in a given lineage. So if you imagine 3 different lineages, for the sake of understanding PBS, imagine that these are 3 different populations. So population A, population B, population C. And one scenario is that population A has had not so much evolution, and another is that there's been lots of evolution along population A, which means that genetically they become more distinct from populations B and C because there's been more genetic change. And PBS is essentially a measure of this distance, the amount of genetic change that's occurred since population A separated from population B. In our case, so this doesn't quite make sense for comparing resistant and alive, because for any locus where it's not associated with resistance, then actually populations A and B are the same population, whereas PBS is sort of dealing with things that should be always separate populations. But it has been used effectively before to detect resistance, association, and so we applied it here as well. And essentially what it does is it uses not FST itself, but a transformation of FST to estimate the amount of distance between A and B, shown here in red, the FST between A and C to estimate this distance in blue, and the FST between B and C to estimate this distance in yellow. And then essentially if you take red, so if you take red plus blue plus yellow, then all of this cancels out. You end up with twice this value. You divide that by 2 and you get your estimate of PBS. And finally, we did a windowed version of the GWAS, where instead of looking for individual SNPs that were super significant, we clustered them and we said we looked for regions that had more significant SNPs than you'd expect by chance. And essentially, I just want to give you a very, very quick overview, because what we essentially found was that in different populations, you get very different hits. So CYP6AA1 was consistent, CYP6AA1 was consistent across different populations, both in Kalutsi and in Anopheles gambiae. But we're also finding TEP genes, which are traditionally involved in immunity. Other cytochrome P450s like CYP9K1 very consistently coming across in all populations. But interestingly, this signal was never bang on CYP9K1. It was always slightly to the left or slightly to the right of it. As I say, other cytochrome P450s, NADH dehydrogenase, which is a cofactor of cytochrome P450s. It's required for cytochrome P450s to work effectively. So lots of different hits in different populations. And we see the same thing with pyrimidine methyl. So that was deltamethrin. This is pyrimidine methyl. Again, cytochrome P450s. CYP9K1 coming out again. A target site of neonicotinoids, insecticides, but one that's still associated with the molecular pathway of ACE1, and so intriguingly might indeed be associated with resistance to PM, but hasn't yet been, there's no previous evidence that this is the case. Some carboxylesterases, which since this paper, we've actually demonstrated that this carboxylesterase cluster here is associated with resistance to pyrimidine methyl, which is some nice functional validation of some of the things that were coming out of this agnostic screening. And then GST2, which is another gene that we see associated with resistance in the literature. And so the take-home message here really is that resistance is really highly multigenic and multi-allelic. So we're seeing different genes involved in resistance in different regions of, in different populations that are still all in West Africa, so very close to each other. And even in loci such as CYP6AA1, which are consistent, we're seeing lots of different resistance alleles. And this is something that we're seeing more and more in all of our analyses. In CYP6AA1, we just keep finding new mutations that are associated with resistance in different populations. And so we need to think about how to deal with this when we're designing wet lab and computational methods for predicting resistance. I don't know why the background's gone blue here. That's super weird. But essentially, metabolic resistance is really tricky to monitor because, to change the expression of this gene, there's a promoter. That promoter binds to polymerases and to transcription factors. There are enhancers. There are other transcription factors that might bind to those enhancers. And if you want to change the expression of that gene, you could mess with the enhancers, you could mess with these transcription factors, you could mess with the promoter, you could mess with this other transcription factor, or you could just increase the copy number of the whole thing, which is what we're seeing with the CYP6AA1 gene duplications. And so there's so many different ways to skin that cat that there's so many different mutations that could lead to resistance through overexpression of that gene. And so we need to think about what we can do about this. So we need to think about, for example, do we need to start doing transcriptomic or proteomic surveillance? So rather than looking for mutations where you're potentially going to be looking for a different mutation every single way you look, does it make more sense to just directly monitor the gene expression? But this comes with all sorts of its own challenges, notably the fact that gene expression is really hard to measure consistently across different regions. So it's really hard to measure gene expression in one region and compare it fairly to a different study measuring gene expression in a different region. We could also try and think about methods that are based on identifying the presence of something going on in a region of those importance. Can we screen a population and say that there is something going on in CYP6AA1? And obviously, I've shown you here that we can with whole genome sequencing data, but ideally, we potentially would like to do that with a cheaper method so we can roll out sort of more broadly. And so is there a way that we can try and see if there's something going on, quotes, in CYP6AA1 just by screening a new population with something more sort of accessible than whole genome sequencing? And finally, coming back to that point I made earlier about sample sizes, so we did some simulations, and we found out that in order to detect those mutations in those associations with KDR and with CYP6AA1, using a GWAS, so using an agnostic screen across the genome with false discovery rate control, we estimated that this could be done with reasonable power, still not huge power, but we're talking sort of 60–70% power if we had a sample size of 500 mosquitoes, so 250 dead and 250 alive, unrelated mosquitoes. So with that, I want to thank everybody that was involved in this study from the GARD partnership and from Malaria Gen, Oxford, Sanger, all of these people, all of the funders, and you for listening. And I'm happy to answer any questions.
Okay, thank you so much for a brilliant and well-explained presentation, Dr. Lucas. We now have we have about 15 minutes for question and discussion. So please type your questions in the Q&A box or the chat box or raise your hand. For the moment, we have about 3 questions already lined up. From Madi Safapour, thanks for the great talk. May you please explain more about sample collection method you used to collect the mosquitoes? Dr. Lucas, that's from Madi.
Yeah, so I didn't do the sample collection myself, but essentially the partners in Africa went to collect larvae. So they were larval collections, identified larval breeding sites in regions that they knew had insecticide resistance, that they'd sort of from previous studies knew that there was resistance in these sites. Collected larvae from pools, brought them back into the lab, raised them to adulthood in the lab, and then exposed them to the various insecticides to find out what the resistance profile was, to establish the lethal doses for that particular population, and then conducted the bioassays. Was there other information that you wanted on the sample collection, or does that answer your question?
Madi, please do type in the chat to answer Dr. Lucas. I'm going to move on to the second question from Mahamat Gaji. The question says, hello, I'm Mahamat from Cameroon. Thank you, Eric, for this very nice presentation, and congratulations on your recent published paper in Nature Communication. I would just like to understand on what basis you chose the windows of 1,000 SNPs to calculate the FST values presented on slide 30. I asked this question because the FST plot shows a large number of signals spread over the whole chromosome, and we can't really select the peak of interest, except for ACE1. Don't you think it would be a good idea to zoom out to reduce the noise, or is there another approach to solving this problem?
Yeah, so that's a very good question. When you're choosing window size, you're essentially trying to balance reducing the noise against being able to home in on the region that you're actually interested in. And the bigger the window you use, the harder it is to then go in and say, okay, well, this is the bit of that region that's important. So yes, so there were a lot of what could have been peaks across the region, but then I hope you saw that with that method of peak detection, then actually a lot of that turned out to be baseline, and what we ended up with was not that many peaks. Once we applied that algorithm in that plot I showed you, there were 4 peaks. In that second plot I showed you, there was only one region that was considered to be a peak. So in sort of playing around with different parameters, we sort of decided that 1,000 SNPs worked fairly well. The other thing is that the bigger the window, the more this law of averages applies, and the more you get this problem of some peaks standing out just because they're under selection, not because they're actually associated with the insecticide. So again, you're trying to balance those two things.
Okay, thank you so much, Dr. Lucas. I see Mohammed has raised their hand. Mohammed, do you have a contribution further to your question?
Yeah, thank you. Thank you, Shoke. Good afternoon again. So just to ask another question on the slide 68, here you mentioned the sample size. Yeah. 68.
Oh, 68, sorry. Yeah.
Yeah, you mentioned actually the sample size to use in order to detect association, a variant associated to insecticide resistance. So I'd like to ask if, for example, in field populations, there are certain alleles that were actually selected to fixation in the field. Do you think that by using this large sample size between the alive and dead when doing the genome-wide association study, do you think that if certain alleles or haplotypes are already been selected in the population to fixation, for example, do you think that by using this huge number of dead and alive, we'll be able to detect the significant allele? Because if the variant is actually fixed, it means that the variant will be actually present in the alive and even in the dead phenotypes. And I think it will blur the analysis or the detection of significant mutations or any other variant that could be linked to insecticide resistance.
Yeah, no. So if the allele is fixed in the population, there's nothing you can do because fundamentally you don't have anything to compare against. So if the allele is fixed in the population, your sample size could be infinity and you still won't detect anything. This sample size calculation was based on the allele frequencies and the effect sizes that we saw in those two significant associations we found when we looked at targeted mutations. So the KDR and the CYP6AA1, we used the allele frequencies that we saw at those loci, and we used the effect sizes that we saw at those loci to say essentially what sample size would we have needed in order for those two loci to have been significant, to have had a significant association. Whether or not that we are absolutely not saying that with a sample size of 500, you will detect every resistance important SNP in the population, because it will depend on the frequency of that SNP in the population. It will depend on the effect size of that SNP, which may be larger or smaller than the ones that we had here. It will depend on all sorts of things. So we're absolutely not saying if you use a sample size of 500, you will detect your associations. We're even not saying if you use a sample size of 500, you will detect all of the associations that have the same effect size and SNP frequency that we found, because as I said, what we found was that the sample size of 500 would have given you a power of sort of 70-ish%. So you're still missing a third of your SNPs that have the same effect size and allele frequency as the ones that we were using. So yes, no, you're absolutely right. I mean, if a SNP is fixed in the population, then you won't find it, no matter what method you use.
Okay, thank you so much.
No problem. Thank you for your questions.
Thank you, Mohammed, for your questions. Okay, so just to remind you, we have about 6 minutes to go. So you do have time to ask questions. Please raise your hand or type in the Q&A or chat box. I'm going to move on to the next question from Sophie Moss. We're saying, great study and explanation. Thank you. What did you use as population C for the PB statistic, assuming A and B are resistant and susceptible populations? Thank you.
90% sure it was a population from AG1000G. So it was a population of anopheles from AG1000G, and I'm pretty sure that it was Mali, which we used as a population, which was slightly separate. West Africa, so different but not too different. So West African, but not in the same countries as the ones that we were studying. It shouldn't make too much of a difference, because whatever you use should be, because A and B are essentially almost identical in the regions that are not associated with resistance, then whatever outgroup you use will probably be different to A and B, and not more similar to one than the other, to your dead than to the alive. So yeah, I mean, it would have been good if we'd been so inclined to spend the time to do it. It would have been good to try different outgroups and see whether the signal was consistent across all of those different outgroups. We didn't really do that. We just stuck with the one and figured that it would probably be fine.
Okay, thank you so much. So there's a question from Joseph Mwangi. It says, hi. Thank you for that informative presentation. I'm Joseph from Kenya. In your research, were you able to determine the rate of mutation in the samples determined to be mutants? Thank you. That's from Joseph.
Sorry, can you repeat the question, please, Ballo?
I'm Joseph from Kenya. In your research, were you able to determine the rate of mutation in the samples?
The rate of mutation. So the allele frequency, yes. The rate of mutation, no, because we didn't have different generations. So we didn't have. I mean, you would need an enormous time, because de novo mutations are extremely rare. And essentially, if in order to get an idea of the rate of mutation, you would have to know what the selection coefficient was for the mutation in order to know how quickly it would spread once it's arisen. And then you would need to ideally see it arise multiple times. So calculating the actual rate of mutation is extremely difficult. But in terms of, so no, we didn't have that. But allele frequency, yes. So for each mutation, we could calculate the frequency of that mutation in each of the populations.
Thanks, Dr. Lucas. There's a question from Cyan Wackley. I hope I'm saying your name right. Hi, thanks for a great presentation. Could you tell me if there was any optimization done to attempt to control bacterial contamination, and if that could help give your data more power? Would raising F1s in the lab help control bacterial levels? So those are two questions from Cyan.
Yeah, I don't know, but we don't actually know what the origin of the bacterial contamination was. We don't know if it was post-mortem or pre, or probably not pre. But we don't know if it was after the samples were collected, after the DNA was extracted. The fact that it segregates by plate suggests that possibly it happened post-extraction. But it's also possible, because I didn't do, and where the person who did do the actual extractions has left, we don't know whether they were done in bags. So maybe that first half of the first plate came in one bag. In that case, possibly it could have been pre-extraction. And the reason it segregates in the plate is because that whole left side of the plate was one bag of mosquitoes. So because we don't know where it came from, we don't know what the cause was. All I would say is I would try and treat all of your samples as identically as possible. I would keep a record of everything, what goes in what bag, and if you have groups of samples in different bags, record which samples are in which bag, so that if you do have a problem, you can try and go back, which we weren't able to do because we didn't have those records. You could go back and find out, okay, it was that bag was contaminated or something like this. I would also, as much as possible, try and treat your dead and your alive samples the same. And I know that's really difficult because obviously you do your bioassay and then you come back, and when you collect them, well, your dead ones have been dead for a while. They have to have been, and your alive ones are still alive, and you kill them immediately. So whether, post-mortem differences in bacterial contamination could be caused by some samples being dead for longer than others is a problem, but it's one that's very hard to address because you can't just go in, you can't be watching and then as soon as the mosquito dies, you take it and you put it away, right? So, but as much as possible, try and treat your dead and alive samples the same.
Okay. Thank you so much. So I see there are about 3 hands raised, and I'm looking at the time as well, so we're gonna have to be quick.
Yeah, and I have another meeting after this one, so I'm afraid I only have time
quickly,
for one more question.
Joshua, last question. Can you please unmute yourself and ask? Joshua?
Hello?
Hi, go ahead.
Thank you for the good and the informative presentation. So my quick question was, do you think because we have not seen the mutation from the Tanzania, do you think the mutation of Anopheles gambiae from West Africa could work this from Gambia? Because I'm not sure if we have Anopheles colusi. Thank you.
Yeah, so I don't know. It would have to move a long way. So what we see from KDR is that there's lots of different origins of KDR, and we're just now seeing the West African F1 KDR move just starting to creep into Uganda now. So it takes a long time for the mutations to go that far. But yes, I think eventually they will. But we have so many different mutations in CYP6AA1. We see different ones in West Africa, different ones in East Africa. I think it's going to be a while before it settles down and maybe one dominant mutation sort of comes to the fore, and it might never quite settle down if we keep trying to hit with different insecticides so that different mutations have different benefits to each of those insecticides. But even in eastern Uganda, I'm not going to go into it now, but we're seeing other TNVs in CYP6AA1 reaching very high copy numbers, and so even that Ugandan one may get replaced by another one soon from East Africa again.
Okay. We've run out of time, so thank you so much, Dr. Lucas, for your time and for a brilliant presentation. For those of